Afternoon, everyone, once again, and I hope you are doing well. Thank you for your patience. So, on behalf of Advent Informatic, I warmly welcome you to today's insightful webinar on Reactive by Design Mastering Covalent Docking with MolSoft ICM-Pro. We're delighted to have you with us today and look forward to a session full of practical demonstration, expert insights, and interactive learning. And thank you for taking the time to join us. So before we begin today's session, allow me to briefly introduce the organization behind this event, which is Advent Informatic Private Limited, headquartered in Pune. Advent Informatic is a leading a distributor of advanced life science software solutions. Our core expertise spans chemoinformatics, molecular modeling, medicinal chemistry, chemical data curation, and biological information management. We serve a wide range of clients, including leading pharmaceuticals and chemical companies, CROs, which is contract research organization, academic institutions, and government R&D labs across India. So our mission is to bridge the gap between, you can say, complex scientific challenges and the advanced software solutions by enabling researchers with the world-class technologies for efficient discovery and innovation. We proudly represent globally recognized technologies partners such as MolSoft, Chemexon, LCVR, Campass, Certara, Transgene IT Solution, and KeyModule. It is now my pleasure to introduce our speakers for today, Dr. Shamshir Singh Sardar, a Senior Application Scientist at Advent Informatics Private Limited. Dr. Shamshir is a seasoned expert in computer-aided drug design, which is CAD, with over 20 years of experience in the pharmaceutical and life science industry. Currently serving as a senior application scientist at Advent Informatic Pune, and Dr. Singh brings a deep expertise in the application of CAD tools to support drug discovery research. With his extensive experience of molecular modeling and computational drug discovery, Dr. Sardar is the perfect guide for our today's session, which is on Reactive by Design, Mastering Covalent Docking with MolSoft ICM-Pro. Before we move forward, during this session, if you have any questions, please feel free to type them in the chat box. We will address them during the Q&A session. So please join me in welcoming Dr. Shamshir Singh Sardar. Dr. Shamshir, the floor is yours.
Thank you, Shailesh. Good afternoon, everyone, and thanks for joining this session. And as we all know that today's session, Reactive by Design, Mastering Covalent Docking with ICM, MolSoft ICM-Pro. So as there is some redundancy in this thing, in the first slide, which already Shailesh has introduced our company, but I would like to say something about our company as well. So we are Advent. We are a 10-year-old company, and we are founded in 2016, and we are based at Pune. And what do? We are the prominent distributors of various life science softwares and databases for drug discovery and medicinal chemistry. You can see in the right, MolSoft, which is a molecular modeling suite, which we are the distributor of the MolSoft. And you can also see Chemexon, which is a prominent leader in chemical structure sketching, editing, property calculations, and all that. Elsevier, again, we are into databases. Like you can try our MBase, MBiology, Reactive, and Pharmapendium from Elsevier. And the SCM, if you wanted to do high-end quantum calculations, you can try using SCM. Campass is again the tool which you can predict the forward reaction mechanism using Campass. Certara is a D360 platform for data visualization, and this is again Transgene, which is an Indian-based company which provides the ELN and other laboratory notebooks as well. So we are again the distributors of all these databases and the tools. So what do? We are again not the developers of these tools. We are the distributors. And what do we do here? We provide the first level of technical support to all the products which we distribute. And 50% of our employees here are PhD holders. And we are highly appreciated by our clients, which by means we recently ran a survey with regards to responsiveness. So most of our customers, they rated us like how quickly we respond to your queries. So like 50% of our customers said like we respond to their queries or their emails in less than 1 hour, and cumulatively 85% said like we respond to their emails in less than 3 hours. So overall in less than 24 hours, we acknowledge that we have received your email and we are trying to work on your issue. So mostly we try to solve the problem either by phone call or by setting up a Teams meeting, and or a MS Teams or a Google Meet, whichever the possible way, and we try to solve it as quickly as possible. If it is something related to code, then that might take a little longer than expected. So otherwise, we try our best to solve your queries in less than 24 hours. And our support is our IP here. So again, this is a complete landscape of MolSoft. To many of you, if you have not heard about MolSoft, MolSoft is not a new company. It's a 32-year-old company. It was founded in 1994. So you can see MolSoft was founded, and there are two technologies based on which the MolSoft runs. One is the internal coordinate mechanics and the biased probability Monte Carlo method. So these two technologies are the backbone of this software, and we also took part in certain challenges like the docking challenge in 1995 and lysozyme antibody prediction. And in the same year, in the same decade, you can see the earlier adopters of this technology were BMS and Genomic Institute of Novartis Research Foundation. In the next decade, again, we partnershiped with Structural Genomics Consortium for developing SCARAP, and we also developed something called Reactive ICM, which is a data visualization tool, which is a small plugin where you can embed this I act, you know, ICM active ICM plugin to your presentation, and you can uh, render the things or import your projects, and you can get the data like in 3D during the presentation as well. And we also partnership with Novartis. It was a 8-year long collaboration, again, where we developed a tool called ICM 3D ligand editor. This was developed mostly for the medicinal chemists, because medicinal chemists, no, they don't want to take out the ligand out of the pocket, and they wanted to do all kinds of modeling studies in the protein pocket by itself. So for them, this is the tool which was developed, and the publication has also been, the work has been also been published in JMC. And in 2008, we also had something called developed a tool called atomic property fields, which is a pharmacophore or a phase in other tools. And we also took part in GPCR challenge. So once the very first in early 2000, maybe in 2003 around, the after the crystallization of the very first GPCR structure. So the crystallographers threw a challenge to build a homology model. Either it can be using the tool whichever you had. So MolSoft tried to build the first adenosine receptor model, and it was ranked top as a top commercial tool in predicting the GPCR model. And we also took part in the Capri challenge, which was more like covalent protein docking challenge, and we were ranked first as well here. And in the next decade, you can see we also launched multiple other modules like for deep learning based mole screen for ADMET, drug repurposing, other things. We also launched GigaSearch, sorry, launched in collaboration with Inamin. And there are many more success stories like FDA approval of Webomere. And we also took part in 3-tier challenges. And Novartis again used the ICM MolSoft VLS on Amazon Cloud, and they used 30,000 cores on Amazon Cloud, and they could finish the computation. They could dock millions of compounds in less than 10 hours, otherwise, which would take like around 39 years of computation time. So, and again, there is a comparative study which we were ranked first in the Covalent docking performance. And in the current decade, as you all know, let me So as you all know, this is a decade of GPUs as well as the AI. And in this decade, we launched something called Write, which can screen. It's a ligand-based screening tool which can screen up to 1.5 to 2.5 million conformers per second per GPU. We also integrated a docking scoring function, AI-based scoring function called radial topological convolutional neural networks. We also brought in another tool called Reach, which is GPU-enabled docking, which can dock up to 100 compounds per second per GPU. This is not a synthon-based or a fragment-based docking. This is a fully brute force, full molecule docking. That is something called, we have a tool called Reach. And Giga Screen, which is again AI plus GPU enabled screening, which can screen up to 1 billion compounds per day. We again also have other tools like CombiReach, Ginger for confirmation generation, and de novo tools have also been launched, and Giga Screen version 2 also been launched in the current decade. And this is again a global subset where you see lots of big companies, global companies, starting from Pfizer, Novartis, Lilly, Vertex, Merck, and many big companies are using MolSoft for their research. And the good part here is this thing, Arrakis Therapeutics, whose pipeline is completely based on RNA discovery, but still they use ICM MolSoft for their research. Similarly, Aravinas, as everyone knew, very recently, the PROTAC has been approved for estrogen receptor, and they are also using ICM MolSoft for their research. Coming to not-to-profit organizations like NIH, NCI again uses ICM MolSoft, and other big universities, global universities like Stanford University, Duke, University of Toronto, University of Southern California also uses MolSoft for their research part. Coming to Indian Institute, how about the Indian users? You can see Syngene, all big CROs starting from Syngene, Aragen, and Origin Oncology, Origin Pharma, Think Molecular, Jubilant Biosys, PI, and Zydus. So these are all the different companies, CROs, as well as the companies are using MolSoft for their research. Coming to the academics, we have very prestigious institutes like Naipar Mohali, Naipar Guwahati, even IIT Gandhinagar, IIT Dharwad, National Institute of Immunology, and couple of central universities like Central University of Punjab and Kerala, Geethams, and many more other colleges as well who are using MolSoft for their research. So, I mean, if you might not have heard about MolSoft, the first question in the mind of the researchers would be like, okay, if I do my research or carry out my research using MolSoft, can I publish it? Yes, definitely. You can see the work carried out using MolSoft has already been published into Nature Cell Death and Disease here, and this is a Nature by itself, which has an impact factor of 70 to 70 plus. And this is again Nature Scientific Reports. This is Nature Cell Death and Disease, Nature Proceedings, Blood, and Science. So yes, definitely, if you carry your research using MolSoft, you can definitely publish into very, very reputed. Apart from these journals, a lot of journals, a lot of medchem journals into which the already work has been published. Like you can see multiple JMCs, EJMCs, BMCs, BMCL, Springer, and many more. Yes. Coming to the modules. So unlike other tool, MolSoft doesn't believe in multiple modules. So just two modules are good enough to carry out your research. The one what we have is ICM-Pro plus homology modeling and ICM-Pro homology modeling plus VLS. So these two modules are good enough where you can, starting from your hit identification till the lead optimization, you can use ICM also. These two modules are good enough to carry out all the activities like docking, QSARs, and everything can be done, which we would be seeing in the upcoming slides. And the other independent modules, which you can again, you know, license out, the ICM Chemist and ICM Chemist Pro are also available independently. So ICM Pro homology plus VLS. So this is what ICM Pro. What are all the different docking studies which you can carry out using ICM Pro? So coming to docking, you can do small molecule docking. Again, you can do protein-protein docking and induced fit docking, which is again explicitly, classified into three different methods. The first one being 4D docking, where you can create an ensemble of multiple crystal structures available, or you can create an ensemble with the various conformations which you can generate during molecular dynamics, and you can create an ensemble and you can run the docking. I mean, you can generate a single grid and dock all your compounds against those maybe 10 or 15 different same protein with different conformations. So coming to hybrid partially explicit, this is, for example, in case of, let's assume in case of kinase, you have a crystal structure which is DFG in, and you have a molecule which is DFG out. But if you dock the molecule type 2 inhibitor into the DFG in conformation, you will not be able to do that. So in that case, you can select a specific residues, like maybe 4 to 5 residues, and you can explicitly generate multiple conformations around that loop, and you can try docking your molecule. So in otherwise, you may have to build a homology model quickly, but this can be done with the hybrid partially explicit methodology available. SCARE is again scanning of dual scanning of alanine and refinement method, where let's assume if your pocket is completely hydrophobic, to understand the ligand binding, how it binds into that pocket. So you can try using SCARE, where it replaces a couple of amino acids in the pocket and docks your molecule. And you can also do RNase protein small molecule docking, and we have integrated for molecular dynamics OpenMM. We also have something called PROTAC modeling. You can do homology modeling, ICM 3D ligand editor, and other, like 2D, you can generate 2D interaction diagrams, protein modeling, electrostatics, bioinformatics tools, predict the effect of mutation using Mercy algorithm and direct database search. This is one of the very interesting feature about MolSoft, which is already integrated with certain biological and the small molecule databases you can see here. So it has been already integrated with PDB, NCBI Blast. If you want to do the homology modeling, you do the basic local alignment search tool, NCBI Blast, which is again integrated. You can search the UniProt, PDB by sequence pattern, sequence length, so all these things. And again, PubChem, Chembel, and certain other drug central databases are also integrated into the MolSoft. You can see here. Yeah. Coming to VLS. So you can again do in VLS, the only difference between Pro and the VLS is in Pro, you can dock max to the max of less than 100 compounds. But here in VLS, you can dock millions of compounds. Like you can carry out structure-based VLS, fragment-based, APF-based VLS, as well as the ligand-based VLS. And again, you get a physics-based scoring function and also an AI-based scoring function, which we call it as RTCNN. And again, as today's topic is mostly on covalent docking, so if you wanted to do or you want to execute covalent docking, then you may have to have this ICM Pro plus VLS license to carry out this covalent docking. And again, you can do 3D pharmacophore search. You can calculate the ADMET properties, various neural network classification, neural network regression, 3D pharmacophore, 2D fingerprint models are available for ADMETs, and you can do hierarchical clustering, 2D, 3D QSAR, MPO, PCA, and various other SAR analysis like R group decomposition, match pair analysis, and many more things. So now let's quickly jump back to the covalent, our topic for the today. So what are covalent inhibitors? So covalent inhibitors are the drugs or which consist of a specific electrophilic warhead that binds to the protein residues, specific like the cysteine, serine, and lysine, which warhead is a electrophilic and this is a nucleophilic kind of reaction which forms a covalent bond between them. And as we know, covalent drugs or covalent inhibitors is not a new research, and serendipitously a number of already known covalent drugs are available. One of them being the aspirin, you can see. So here, aspirin, as everyone knows, this is one of the oldest medicines as early, which was introduced in the early 19th century. So it acts irreversibly by inhibiting the cyclooxygenase 1 and 2 by forming a covalent acetylation of the serine residue, thereby reducing the production of prostaglandins and thromboxanes. As we all know, aspirin is very commonly used to relieve pain, fever, inflammation, and but also it has multiple other usages also. At a lower doses, it also prevents blood clot formation, making an important therapy for reducing heart risk of heart and stroke. So this is regarded as one of the earliest and the most successful examples of covalent drug. Coming to the second, the penicillin, which is the one of the very first uh, blockbuster antibiotics, important discoveries, and it works by covalently binding to the bacterial enzyme responsible for the building the cell wall and prevent the bacteria from forming the strong protective layer. Nothing but they are cell wall inhibitors. So ultimately, what happens? The bacteria ultimately kills the bacteria, leaving the human cells unaffected. So penicillin not only revolutionized the treatment of bacterial infections, but also established a covalent inhibition as a powerful strategy in drug discovery. So there are again other drugs like omeprazole, which is an antacid, forms a covalent disulfide bond with cysteine, and clopidogrel, which again covalently and irreversibly binds to P2Y12 receptor on the platelets and prevent clotting. So this is again from serendipity to precision, the evaluation of the covalent drugs. You can see as early as in 1890s, the aspirin was discovered, and since then the penicillin followed by omeprazoles, and again these two lansoprazole. This is an anti-infective drug which is phosphomycin and clopidogrel, bortezomib, which is again an anti-cancer drug, saxagliptin, which is again anti-diabetic drug. So in from 2010s, since this 2000 saxagliptin, which is an anti-diabetic drug, many rationally designed targeted covalent inhibitors have reached the clinic. For instance, you can see this ibrutinib, which was approved in the year 2014 for as a BTK inhibitor, and the successors of this, like you can see, acalabrutinib, zanubrutinib. So these were again also approved for BTK as BTK inhibitors. Similarly, EGFR inhibitors like afatinib and osimertinib and dacomitinib, which are again mutants against NSCLC cancers. And apart from these, even in 2011, there is another drug which was uh, approved for as an HCV protease inhibitor, teleprevir, which is approved in 2011, and again KRAS G12C glycine to cysteine inhibitors. There is a drug called sotorasib and adagrasib, which are being approved. The very well-known covalent inhibitor, which for the COVID we call it as Paxlovid, which was again approved in 2021. So this is a targeted antiviral um, medicine developed to treat COVID-19 by blocking the MPRO protease. So this is which is essential for viral replication. So what it does, it forms a irreversible covalent bond with the catalytic cysteine in the protease and providing strong and selective inhibition with again minimizing the off-target activities. So this is how this complete journey has been going on till today. Again, you can see the nucleophilic warheads, nucleophilic amino acid residues, and their mechanisms. As we know, covalent bond formation occurs between a nucleophilic amino residues and reacts with the electrophilic warhead of the inhibitor. So cysteine residues most commonly, you see cysteine residues most commonly form or undergo Michaelisation with acrylamides, vinyl sulfonamides, and vinyl sulfones, sorry, and carbonyl groups and to form thiohemiacetyls, and also open epoxide rings. So serine residues can react with certain electrophiles like boronic esters and form tetrahedral adducts, and also certain lysine groups to form Schiff's bases with the aldehydes and undergo nucleophilic substitution with aryl fluorides. So these are all these are two different types of reversible and irreversible covalent inhibitors. Again, most covalent inhibitors act in a two-step mechanism. Similarly, uh, in MolSoft also the docking approach is a two-step. Firstly, the inhibitor binds reversibly to the target, forming a non-covalent complex defined by the equilibrium of Ki. This is followed by the covalent bond formation again, which is again characterized by rate constant called Ki inert. This is Ki inert, and in and irreversible inhibitors, the reverse reaction K minus 2 is negligible, resulting in the permanent target binding. The efficiency of covalent inhibition is expressed as the ratio of K inact and Ki ratio, where the higher values indicate the more potent inhibitors. And these are all the different amino acids which take part in covalent binding. Like you see cysteine, arginine, tyrosine. The side chain of tyrosine OH can takes part in the covalent bond formation. Serine OH and threonine, and these are all the different residues which can take part in the covalent bond formation. So these are the representative covalent warheads and target nucleophiles, reactivity, and their stability. You can see that acrylamide, which targets the residues called cysteine, and the kind of reaction what we call it as a Michael addition, which is a moderate to fast kind of reactivity. It is a very well studied and installed on the scaffolds, and irreversibly, which is again already studied. A lot of compounds are already in the market available, as I said, ibrutinib and certain other afatinib drugs available for EGFR and BTK target uh, covalent inhibitors. So off-target reactions in this scaffold binds weakly, irreversible toxicity concerns. So vinyl sulfonil or sulfonamide, which is again a cysteine, which again works on Michael addition. This is again a very fast reactive. Haloacetamide, which again targets cysteine. So nitrile again targets cysteine and serine. So this way, these are the very well known warheads which are known in literature, and these are all the very well known amino acid residues and the kind of reaction they take place, and the reactivity with regards to speed moderate to fast. So this tells us about what are all a complete landscape about the different warheads and that kind of amino acid residues and the kind of reactivity that they possess. Yeah. So this is again one of the success stories. You know, Rampex Pharmaceuticals, they launched a drug in the year 2017 called Vivomir, which is a covalent inhibitor, beta lactamase covalent inhibitor. So this particular molecule, we can claim that the compound, we have the authority to claim that uh, this particular molecule which has been approved, it was designed using the structure based methodologies from using the ICM MolSoft. This is another success story, comparative evaluation of covalent docking tools, where this is the Hungarian group of Academy of Sciences, where they compared different tools available in the market with regards which can perform the covalent docking, starting from Autodock and CoveDock fitted and MOE. So you can see with an RMSD of cutoff of 3 also, MolSoft performed much better. And ICM outperformed with all other programs in the covalent docking test and reproducing experimental binding modes. And it performed very well with more challenging flexible ligands as well. So let's get back to the covalent docking workflow in MolSoft. How is this workflow? So what all you need to do is you have to, a typical way of docking, import the protein, convert the protein, nothing but prepare the protein, converting it into a suitable tool, and then you select the ligand or you generate a grid around that, and then followed by you set up a reaction mechanism, select the reaction mechanism, and then you dock the compound. So this is how the complete workflow happens. So, but again, as I said, even in MolSoft, the covalent docking is a two-step method where ICM covalent docking is modeled by defining the chemical reaction that transforms the free ligand and the target receptor side chain to be unified or covalently linked as a product. So to simulate this process efficiently, software masks the reacting side chain of the specific residue during the computational reads. So we will see this during the demonstration, how we mask the side chain of that specific residue during the computational reads. So upon introducing the input library, automatically ICM applies the chemical transformation and enforce geometric constraints and designated side chains during a Monte Carlo simulation, docking simulation. So which again results in the binding poses, which reflect the accurate and low energy physical conformations. So let's quickly jump back to the yeah. Before that, docking, there is something called prediction of reactive cysteine. We will also try to understand how you can utilize the understand whether the cysteine which you wanted to target is it really reactive or not. Again, it depends on the closest proximity of the residues nearby the cysteine. We will try to take a protein from the COVID, and we will try to calculate the reactivity of the cysteine, then followed by we will do the covalent docking as well. So this is the an empty GUI where you can see where I'll try to import a PDB called 6Y2E. So this is the crystal structure of the SARS-2 COVID protein. And now you see this protein doesn't have any uh, ligand bound, and there are multiple cysteines available in this protein. So what I'm trying to do is I'm trying to load them firstly. So these are all the different cysteine residues that are there, and I just wanted to identify or understand which of those cysteines should I target for my for my research. So now what I can do, I can quickly go to tools, 3D predict, reactive cysteines. So these are all the residues which, if there is a ligand also, you can exclude the ligands as well here, but there is no ligand in this protein. Now these are all the 12 residues, and I want to calculate the reactivity of all the different cysteines that are available. So it takes a little time to calculate the reactivity here again. Now you can see the various cysteines are encoded with different this is a table which tells, and the most reactive amino acid what we see here, cysteine is cysteine 145, right? So what are all the different things? This is the residue numbers, and this is again the distance to protein surface. So how much the particular cysteine is exposed to the surface. Again, this is solvent accessible surface area of the cysteine side chain. Here you can see, right? Again, there are two residues here, 44 and cysteine 145. So both these, the pocket area is similar. But again, the differing thing here you can see is the solvent accessible surface area of the side chain, which is very much lower in cysteine 44, and cysteine 145 it is much higher. Maybe this could be one of the reasons. This is one of the reasons why the score is high. So the higher the score, the reactivity. So all these cysteines are reactive, and among them the most reactive cysteine is cysteine 145. So again, this also calculate near n plus. This is something, how distant is the near the positively charged amino acids like lysine and arginine are, because as I said, uh, the reactivity all of the cysteine also depend on the closest proximity of the amino acid residues. So again, yes, all the positively charged amino acid does have some impact on the reactivity of the cysteine as well. And again, you can also see the distance to nearest histidine. What is the distance is 3.6 angstroms, and this is again the electrostatic potential of the the sulfur of the beta sulfur of the cysteine, and this is a hydrogen bond backbone energy. So With this, we could conclude that this particular cysteine 145 is one of the most reactive cysteines. Now what I'll do, I'll try to import another PDB 6Y2F, which is a co-crystallized with a covalent inhibitor. And then I will try to align these two proteins. And again, you can see how quickly and how easily you can align in these two proteins, right? I'll just try to put them here in the workspace. You can see I have selected them, and then one single click, and my reference protein is 6Y2E. I say OK. Now you can see, as per my earlier analysis, cysteine 165 is one of the most reactive. 145 is the most reactive, you know, cysteine. And now you can see here the same case. Similarly, this particular molecule is covalently bound to the cysteine 145. So again, this particular uh, prediction tool is a random forest classification tool, which is again cross-validated with a test set of 20% of the random data. So this is how it accurately predicts the reactivity of the cysteine. Now let's assume you are working on a reversible inhibitor, and maybe you might find any cysteine in the closest proximity of the protein, so that you try to plan to convert that reversible inhibitor to a irreversible inhibitor. And then let we should, before we make those molecules and uh, test them in the lab, again, which is very cost expensive thing. Before that, if you can predict whether the cysteines, whether the, if I can I target that particular cysteine, whether I have the reactivity, you can try using the this tool, 3D predict and reactive cysteine. So once you find some good information about the reactivity, you can start designing your inhibitors with a suitable warheads like acrylamides, vinyl sulfones, and the rest all up to the user, which nucleophilic warhead he wanted to put onto the ligand. So this is how the user can predict the reactivity of the cysteine. So let me quickly jump on to let me open another GUI, and then I'll try to import a protein called 3PBS. So this is again a bacterial protein, which is co-crystallized with a drug called astrionam, right? This is covalently bound to this protein target, right? Now very quickly, after importing this, I'll convert this to an ICM format by selecting these options. So I'll keep tight waters is something where if any of your water molecules are making more than 2 or 3 hydrogen bonds, we try to keep them. This is again delete all waters, removing all the waters, delete loose, which make one hydrogen bond less than 2. So keep all waters is up to the. So again, I'll try to delete all the waters because I knew there is no water mediated, uh, interhydrogen bond forming waters here in the active site. So I'll try to delete all the waters, and we are while conversion from XR structure to the ICM. What it does, it tries to add the hydrogens, and we are going to optimize the orientation of the hydrogens as well. So we are going to optimize the histidine, proline, and aspargine, glutamine, and cysteine as well. We are replacing the original XR with the ICM format. We will display the results, and if there is any missing side chain, if you can uncheck, then it builds that side chain during the conversion. And we are assigning the secondary structure. And MolSoft has an inbuilt predict formal charge. I mean, a PK model which will predict the formal charges on the ligands as well. And this is an optimized ligand tautomer. So it will try to identify whether there is any ketoenol tautomerism with the ligand. So by selecting all these, we click on OK. So now very quickly, this XR structure we are preparing. This is something what we call it as protein preparation. Now I would like to understand what are all the different interactions this ligand is making. To understand that, just one single right click, display pocket and ligand pocket interactions. And now you can see this astrionam drug which is a drug, is making multiple hydrogen bonds with the uh, protein. Now, as you see, they are very, I can't see them clearly. So just by increasing the width of the sphere sizes, you can see this ligand or the astunium drug makes multiple hydrogen bonds, and you can again see the hydrogen bonds in different colors, green and blue. In MolSoft, the hydrogen bonds are represented in three different colors, red being the strongest hydrogen bond, followed by green and blue. So even you can see some cases, the green color, there are sphere size differences in the sphere sizes. This means this particular hydrogen bond is stronger than this one. So this is how you can easily understand the strongest hydrogen bonds here. And even you can again change the notation as per your requirement. It's not just a sphere way, the way of representation. You can change it to solid line, you can change it to dotted line, and even you can change the energy. And here the representation to by energy you can change to telescopic, and even you can increase. So by default, when you click on the hydrogen bonds, the energy, minimum energy it considers is 1 kilocalories. If you are working at any of the metal containing proteins, you wanted to see those hydrogen bonds which are very low energetic, you can try decreasing the energy of the hydrogen bonds as well. So this way, just this GUI has just one single click, one single option has so many ways of representation of your hydrogen bonds as well. And now let me see where the hydrogen bond is, the covalent bond. And now this is the serine 294 is the this with this residue you see a covalent bond here. I'm trying to highlight this with this, right? So now again what I have to do is I have to quickly break this covalent bond and remove this compound from the pocket and generate the grid maps, and then dock this astronome into this protein as well to check whether how efficiently it is able to reproduce the biological conformation of this, and along with the chemical transformation as well. So to do that, what I'll do, I will right click on this ligand, and then I'll just say, okay, move from the object, which we are moving away. Now this is the ligand is not the part of your ligand. Now you see in other tools you may have to adjust various other bond orders and all that, but in MolSoft it automatically takes and it adjusts the bond orders. So let me double click on the ligand and go to docking, new project. This methodology is as same as your small molecule docking, just one step more where you mask the residue of the serine, and then you generate the grid maps, and you make the chemical transformation as well. So mydoc underscore cov is the name of the job name, and I'm clicking on define sites around selected ligands. So these are all the different residues. When I click on this, so it selects the residues which are there in the 5 angstroms. I'll try to off this receptor, make receptor maps, and say okay. Now when I am building this, you see a probe in the middle. So this tells uh, you say the system where to start the simulation. Now we will click on go, and it gives you an external grid box where you can either increase or decrease just by catching any of the corners of uh, these grid boxes. And this is very interactively, the user can either increase or decrease, but in others, like you get in XYZ where you never know, when you change a particular coordinate, your grid box might jump over to somewhere else. When you say on OK, now my grid, the centroid has been defined. Now what I'm going to do is, this is again I need a ligand as well to dock it firstly. So before that, what I will have to do is I have to define the chemical transformation which I wanted. So as I said, this is a drug called astrionam, and let me extract this ligand as well. Right click, extract ligand. Right now, whatever the structure in 2D which you see, this ligand is, there is a 4 numbered ring here which is again opened up, and this aldehyde is making a covalent bond formation with the serine residue. But if we dock the same molecule, I mean, we may not be able to form the covalent bond. For that reason, we may have to, bring the full astrium structure. Again, the user doesn't need to sketch it. Just click on this sketching button, go here and search it with as name, with the name of the drug, AZTREONAM, right? And then I'll add this to the table, right? Now you see what's happening. So this is the key difference between the ring has been opened up here in this case. So now we will try to dock this compound, the four, which has the 4-numbered ring, and selecting the reaction transformation, you will see the molecules after docking, you get this ring gets opened up, and this aldehyde, free aldehyde, forms a covalent bond with the serine 294. Now let's go to docking and do a covalent docking. For that, I may have to select the residue which is involved in covalent bond. Now you can see I've selected this residue and load or preset reactions. So as I know serine, this I'm trying to target the residue called serine. Now what are all the different? This is a there is a huge uh, library of different reaction mechanisms already there. So you can try using with the name, and you can try using with the you know the warhead, and you can try searching it with the kind of reaction which you wanted to take it up. So now, as this being a beta lactam ring opening reaction, I would be very much interested in selecting this reaction. I'll add this to the docking project and say, and we are remaking the receptor maps. Now let's see, as I said, like, we are going to mask the serine. Now when I say okay, you see that this particular residue will not be considered as for the grid maps. When I say okay, now you see this has off. So it means rest all amino acid residues are considered for grid map generation, except the serine for the particular chemical reaction transformation. Now we have our grid maps ready for docking, and we have the chemical structure, 2D structure of the astrium ready in the chemical table as well. So what I'll do is I'll go to docking. Quickly I'll say dock chemical table, and I'll say okay. So again, as I said earlier also, this is again a two-step docking method, where it initially docks the ligand and it tries to form the hydrogen bonds followed by the reaction transformation. So these are other different things. Thoroughness is something how extensively, let's assume if your ligand has more number of rotatable bonds, you can increase this thoroughness for sampling it, sampling it better. And again, you see this pop-up window, my covalent docking job is completed. If I say okay, it will be loaded as a new chemical table, right? Yeah, it has added. So till then, let's discuss about these things. So the number of confirmations what I gave is 3, and sampling double bonds option is again available here. Sample racemic. As you see, this is only one isomer. So if this being a racemic one, you can try selecting this. It will generate both R and S isomers, and it will try to log. Even if you have multiple chiral centers, it generates both RR, RS, SR, multiple enantiomers as well. And if you have any kind of keto-enol tautomerism, you can again sample them here as well. So now let us analyze the docking results. Let me check this chem.dock. Again, to load this molecule, I'll try to off the crystal ligand in the workspace. If I want to load just one single click, so this is how it loads your molecule. Now you can see with one single click here, I'll try to convert into a pretty beautiful view, which you can see here. So I'll try to off the CPK mode, and also I'll decrease the ball and stick thickness ratio for better visualization, right? Let me again change the representation of the protein, right? Even I can make it to secondary structure. So you can see all these modifications which here I am doing. Just by 3 clicks, I could make it a beautiful, way of representation here. Now let me hover, bring my crystal ligand onto that. And now the yellow colored ligand, what you see, little dull colored. So this is a crystal conformation, and again you can see this transformation has already occurred. And like, let's see the same transformation here in the 2D table as well. And you can see here this particular part of the ligand, the keto, the aldehyde part is making a covalent bond with the serine side chain. Though this is a amino acid number serine 294, and these are all the different scoring parameters which are which you usually see. This is a physics based scoring method, what we call it as, you know, the docking score. N atom is number of atoms. This is the number of rotatable bonds, 12, and this is the energy of the hydrogen bond and hydrophobic energy, van der Waals energy, and this is the, you know, internal conformational energy of the ligand. Usually, you can consider it as the strain energy as well. So this is again the dissolution energy of the exposed hydrogen bond donors and the acceptors. The lower, the better. Similarly, this is something what we call solvation electrostatics energy upon the change in the solvation energy, electrostatic energy upon binding, the lower, the better. Again, this is a statistical scoring function. This is a RTCNN, which we call it as an AI-based scoring function. This is the loss of entropy by the rotatable protein side chains. So receptor conformations. So this is usually one, because now we are working with only one protein. But when you are working with an ensemble of proteins, which I explained during the presentation part, where you generate an ensemble of 10 or more proteins in the 4D. So in that case, to whichever the confirmation it binds, it will give you the number here. So the number changes here in that scenario. So these are all the different scoring parameters as well. And this is how, let me calculate the RMSD as well here. Go to tools, 3D predict, sorry, analysis and RMSD. And now this is the same table which I can calculate for proteins as well as the chemicals. So the first molecule being my reference compound, and this is my the dot compound. When I say okay, the RMSD is 1.2. Anything less than 2 is considered to be better. And the reason for 1.2 is some of his side chains, you know, they got flipped over the other side. So this is again in the solvent exposable. This is again into the exposable pocket of the protein. Yeah. So this is all about covalent docking. Uh, with this, I would like to conclude the session. And there is one point which I would like to tell you. For example, if any of the reactions are not available, what you can do is you can either write to us, or you can again write. I mean, you can sketch your own reactions and you can import them and you can try running the covalent docking. So thank you everyone.
Thank you so much, Dr. Shamshir, for that detailed and highly informative session. So now we will move to the Q&A session. Before that, once again, if you have any questions related to today's topic, including covalent docking strategies or the use of MolSoft ICM-Pro for covalent docking studies, so please feel to type them in the chat box. So, Dr. Shamshir, we have already received few interesting questions from our audience, so let's take them one by one. First question is, can we do covalent docking VLS?
Yes, you can do covalent docking VLS.
Okay. So with the continuity of first question, if yes, is there any publication associated with it?
Yeah, there is a publication. I mean, we have a I mean, it was done very old publication we have, where they already performed these studies. They have used for the identifying a small inhibitors against a particular virus. So let me share once again, share you my screen where
Okay.
they, yeah, just a minute. So yeah, this is the article. They used, I mean, they modeled the pox virus protein proteinase using homology modeling, and they also did the covalent docking where they docked a library of 230,000 compounds containing ketone and aldehydes, of which 456 were predicted and 97 inhibitors of 17L protease activity were confirmed in biological assay. Yes, definitely. Within, again, you see here the hit rate, which is 20%, which is very, very high. Yes, definitely you can, you can do the VLS for covalent docking as well.
Okay. The next question is what to do if the reaction transformation is not available in the reaction library?
Yeah, you can reach to us, or you can try writing a reaction by yourself. If you are not expert into that, you can try reaching us. We will try to write or create an reaction transformation for you, and we can share you back the reaction file with you so that you can run your own, that reaction transformation as well.
Okay, great. And the next question is, does the OpenMM successfully runs for the protein ligand covalent complex?
Yes, we tried running one of these covalently docked complex very recently. Yeah, OpenM, there is no issue with the OpenMM MD.
Okay, great. And the other question is, do MolSoft support any covalent docking database for ligand without warheads?
Ligand,
Should I
MolSoft,
repeat?
can you please repeat?
Do MolSoft support any covalent docking database for ligand with warheads?
No, we don't have anything as such.
Okay, that's it. And thank you all for the excellent and thoughtful questions. Your active participation truly makes this session more engaging and meaningful. While we have tried to address as many queries as possible, a few may remain unanswered due to a time limitation. Don't worry for that. Our team will review all pending questions and get back to you via email with the detailed responses. So thank you once again for your participation. With that, we come to the end of the today's webinar. On behalf of Advent Informatics, I extended a sincere thank you to Dr. Shamshir Singh Sardar for sharing his expertise and providing valuable insight into covalent docking methods and the powerful capabilities of MolSoft ICM-Pro in the modern drug discovery. And a big thank you as well to all our participants for your time, for your engagement, and for this session, for your participation. So we look forward to welcoming you again in our upcoming webinar with more insightful scientific discussions and hands-on learning experience. Till that, have a wonderful day ahead. Thank you.
Thank you all. Thank you all. Bye bye.
Thank you so much, Dr. Shamshir.